A disease killed more than 1,080,000 people in the United States. A virus spreads very easily to other people. 659121949 people affected worldwide. Cases confirmed in every country in the world. Coronaviruses are a group of related RNA viruses that cause diseases in mammals and birds. In humans and birds, they cause respiratory tract infections that can range from mild to lethal. Mild illnesses in humans include some cases of the common cold, which is also caused by other viruses, predominantly rhinoviruses, while more lethal varieties can cause SARS, MERS and COVID-19, which is causing the ongoing pandemic. In cows and pigs, they cause diarrhea, while in mice, they cause hepatitis and encephalomyelitis. Coronaviruses constitute the subfamily Orthocoronavirinae, in the family Coronaviridae, order Nidovirales, and realm Ribaviria. They are enveloped viruses with a positive sense single-stranded RNA genome and a nucleocapsid of helical symmetry. The genome size of coronaviruses ranges from approximately 26 to 32 kilobases, one of the largest among RNA viruses. They have characteristic club-shaped spikes that project from their surface, which in electron micrographs create an image reminiscent of the stellar corona, from which their name derives. The name coronavirus is derived from Latin corona, meaning crown or wreath, itself a borrowing from Greek kappa omicron rhonueta corn, garland, wreath. The name was coined by June Almeida and David Tyrrell, who first observed and studied human coronaviruses. The word was first used in print in 1968 by an informal group of virologists in the journal Nature to designate the new family of viruses. The name refers to the characteristic appearance of virions, the infective form of the virus, by electron microscopy, which have a fringe of large, bulbous surface projections creating an image reminiscent of the solar corona or halo. This morphology is created by the viral spike peplimers, which are proteins on the surface of the virus. The scientific name coronavirus was accepted as a genus name by the International Committee for the Nomenclature of Viruses, later renamed International Committee on Taxonomy of Viruses, in 1971. As the number of new species increased, the genus was split into 4 genera, namely alpha coronavirus, beta coronavirus, delta coronavirus, and gamma coronavirus, in 2009. The common name coronavirus is used to refer to any member of the subfamily Orthocoronavirinae. As of 2020, 45 species are officially recognized. The earliest reports of a coronavirus infection in animals occurred in the late 1920s, when an acute respiratory infection of domesticated chickens emerged in North America. Arthur Schock and M.C. Hahn in 1931 made the first detailed report which described a new respiratory infection of chickens in North Dakota. The infection of newborn chicks was characterized by gasping and listlessness with high mortality rates of 40 to 90%. Leland David Bushnell and Carl Alfred Brandley isolated the virus that caused the infection in 1933. The virus was then known as infectious bronchitis virus, IBV. Charles D. Hudson and Fred Robert Baudet cultivated the virus for the first time in 1937. The specimen came to be known as the Baudet strain. In the late 1940s, 2 more animal coronaviruses, JHM that causes brain disease, murine encephalitis, and mouse hepatitis virus, MHV, that causes hepatitis in mice were discovered. It was not realized at the time that these 3 different viruses were related. Human coronaviruses were discovered in the 1960s using 2 different methods in the United Kingdom and the United States. E.C. Kendall, Malcolm Bino, and David Tyrrell, working at the Common Cold Unit of the British Medical Research Council, collected a unique common cold virus designated B814 in 1961. The virus could not be cultivated using standard techniques which had successfully cultivated rhinoviruses, adenoviruses, and other known common cold viruses. In 1965, Tyrrell and Bino successfully cultivated the novel virus by serially passing it through organ culture of human embryonic trachea. The new cultivating method was introduced to the lab by Bertil Horn. The isolated virus, when intranasally inoculated into volunteers, caused a cold and was inactivated by ether, which indicated it had a lipid envelope. Dorothy Halmrott and John Prochnow at the University of Chicago isolated a novel cold from medical students in 1962. They isolated and grew the virus in kidney tissue culture, designating it 229E. The novel virus caused a cold in volunteers and, like B814, was inactivated by ether. Scottish virologist June Almeida at St. Thomas Hospital in London, collaborating with Tyrrell, compared the structures of IBV, B814 and 229E in 1967. Using electron microscopy, the three viruses were shown to be morphologically related by their general shape and distinctive club-like spikes. A research group at the National Institute of Health the same year was able to isolate another member of this new group of viruses using organ culture and named one of the samples OC43, OC for organ culture. Like B814, 229E, and IBV, the novel cold virus OC43 had distinctive club-like spikes when observed with the electron microscope. The IBV-like novel cold viruses were soon shown to be also morphologically related to the mouse hepatitis virus. This new group of viruses were named coronaviruses after their distinctive morphological appearance. Human coronavirus 229E and human coronavirus OC43 continued to be studied in subsequent decades. The coronavirus strain B814 was lost. It is not known which present human coronavirus it was. Other human coronaviruses have since been identified, including SARS-CoV in 2003, HCoV-NL63 in 2003, HCoV-HKU1 in 2004, MERS-CoV in 2013, and SARS-CoV-2 in 2019. There have also been a large number of animal coronaviruses identified since the 1960s. Thanks for watching. Don't forget to like, share and subscribe. Visit our channel for more informative content.